This video is on the topic of how enzymes are affected by the pH of the solution they are in. The structure of an enzyme is very important for its function. Enzymes have a specific 3D structure and active site shape that allows them to fit with their substrate as part of the lock and key model. This structure is held in place by chemical bonds between the amino acids in the protein of the enzyme. The bonds holding together the amino acids in the enzyme are specially adapted to withstand specific pH conditions. When the pH changes to be much higher or lower than this, the bonds between the amino acids can break. When the bonds break, the active site of the enzyme loses its shape and the enzyme becomes denatured, meaning the active site can no longer bind to the substrate and the enzyme cannot catalyze reactions. This graph shows the relationship between the rate of enzyme action and pH for an enzyme found in the small intestine of humans. The peak of this graph in the middle is where the rate of reaction is fastest. This pH is called the optimum pH. In the small intestine, this is approximately pH 8. Above or below the optimum, the rate of enzyme activity decreases because the active sites of some of the enzymes are changing shape in the acidic and alkaline conditions. At extreme pH conditions above and below the optimum, the active sites of all the enzymes become denatured, and the enzyme activity stops. Make sure you're familiar with this graph because you may be asked to draw, describe, or explain it. Not all enzymes have the same optimum pH as each other, and are instead adapted for the conditions of the area of the body in which they normally function. Some examples are shown in the graph here. You can see that pepsin, which is an enzyme found in stomach acid, has an optimum pH of 2 to prevent it from denaturing in the acidic conditions of the stomach, while the enzyme urease has an optimum pH of around 7 and trypsin has an optimum pH of around 8. A common mistake to make is to say that all enzymes denature in acidic conditions. Remember that different enzymes function best at different pH levels. In summary, the structure of an enzyme is held in place by bonds that are affected by changes in pH. When the pH becomes too high or low in comparison to the optimum, it causes the bonds holding the enzyme structure together to break, changing the shape of the active site and preventing it from being able to bind to the substrate. The enzyme is denatured. The pH that allows the enzyme to have the fastest rate of reaction is called the optimum pH, and this varies for different types of enzyme.